My name is Brooke Anderson. I'm a graduate student at UC San Diego in Rachel Dutton's lab. We study the microbiology of cheese rinds. And an important part in studying these microbiomes is to know the kind of molecules that microbes are producing that might affect the growth or the behavior of a neighboring microbial species. Cheese as a milk product is a very complex chemical environment. In the cheese-making process, the milk is fermented by lactic acid bacteria. Then a cheese may be aged to develop a surface rind of bacteria and fungi who are all contributing to odor and flavor compounds in this cheese. So this results in very complex chemistries, and interpreting a chemical profile and linking it back to its biological origins is one of the key challenges in microbiology, as well as in biochemistry, medicine, and other fields. We want to be able to organize, to simplify, and then visualize these complex data in order to make sense of it. And molecular networking is a tool for organizing mass spec data. It creates a network that groups compounds that are structurally similar, and in doing so, it captures chemical relationships. And this means that we can consider chemical families instead of just individual compounds. And you'll see how this is really useful when I show you how bacteria and fungi really contribute to the smell and taste of a cheese. So the cheese that we were looking at in this study is a Stilton, which is a blue cheese. And we were interested in differentiating the molecules in the curd of the cheese from those at the rind. And this was supposed to enable us to look at how rind-associated microbes contribute to the smell and taste of a cheese differently, maybe, from those microbes that are in the curd. And so to do this, we collected data for volatile compounds in chunks of the Stilton using GCMS. And we also collected sequencing data from both the rind and the curd. The GCMS generated a lot of data. There were hundreds of compounds detected. This can be very overwhelming, and it would take a lot of time to go through this data manually to organize it and make sense of it. And so we took our data and created a molecular network. And this organized our data by grouping compounds that shared structural similarities. And this allowed us to really streamline our analysis of microbial activity in this cheese. So here's all of our GCMS data displayed as molecular network. And the first cluster that we're going to look at together is the first cluster that I really looked for in this data set. Since this is a blue cheese, a mold called Penicillium roqueforti was added to the milk, and then the curd was pierced in order to deliver some oxygen for this mold to grow. The characteristic smell and taste of a blue cheese is due to short-chain methyl ketones made by Penicillium roqueforti. And here you can see we have a grouping of these methyl ketones, such as 2-heptanone or 2-nonanone, that show very similar distribution between the curd and the rind, since the Penicillium roqueforti fungus spans through the entire cheese. Another cluster that caught my attention is this cluster of sulfides. All these compounds are end products of the breakdown of the amino acid methionine. The biochemical pathways generating these sulfides from methionine have been fairly well documented in Brevibacterium, which our associated sequencing data indicated was present in this rind. These sulfur-containing compounds can smell like stinky feet, and we frequently associate them with the pungent stench of stinky cheeses like Limburger. Despite decades of biochemical research into the production of volatiles in cheese, there are still areas to explore. This is particularly evident in this one cluster of primary fatty amides, which all show a very similar pattern of dominance at the rind. While butanamide has been mentioned in cheese literature as having a sort of nutty flavor, the synthesis of this group of molecules by microorganisms has not been explored, much less their role in biology or in cheeses. Yet, this cluster indicates that these molecules are being created by microbes specific to the rind, and that there's probably a general pathway for their creation, given this very consistent pattern of rind versus curd for all of these molecules in this cluster. So, as you can see from these examples, organizing GCMS data into molecular networks has allowed me to very efficiently and effectively explore and contextualize the Stilton data.